Okay,
No
well,
problem.
it's my pleasure to introduce Dr. Lara Boyd, a distinguished neuroscientist and physical therapist at the University of British Columbia. Dr. Boyd is a professor in the Department of Physical Therapy, Canada Research Chair in Neurobiology of Motor Learning, and the director of the Brain and Behavior Laboratory. Her groundbreaking work focuses on neuroplasticity and motor learning, particularly in the context of stroke rehabilitation. Using advanced neuroimaging and brain stimulation techniques, she has made significant contributions to understanding how the brain reorganizes itself during learning and recovery. Her work has been instrumental in shaping rehabilitation strategies and optimizing recovery for individuals with neurological injuries. Thank you, Dr. Boyd, for joining us today.
Yeah, thank you so much.
No worries. And to start, could you tell us just a little bit about yourself and what initially drew you to the field of neuroscience and physical therapy?
Yeah, I'd be happy to. So I became a physical therapist thinking I was very interested in sport, like a lot of PT students are. And I really thought that I would go into some kind of sports rehab or sports optimization kind of practice. But as I undertook some training, I became really, really interested in brain damage and brain diseases. I just was able to get a really exciting amount of exposure to patients like that. So I wound up, when I finished my degree, taking an early kind of, what's now called a clinical residency in rehab, but I did an opportunity like that where I went to a major neurologic rehab center in the US, and I became very involved in stroke recovery and trying to help rehabilitate stroke patients. And it honestly didn't go well. My patients, I always felt like didn't get enough recovery. It was really frustrating. I'd worked really hard with them and I felt like the changes just didn't persist. And I finally realized that I felt like I was trying to repair a system that I just didn't understand. So I couldn't make it work. I always say it would be like being a car mechanic, but you have no idea how the engine works, but you're trying to fix this car. And you open the hood and you're like, I have no idea what all this is. Actually, it's how I feel if I open the hood of my car. But that's how I felt about the brain then. So quit my job and I went back to school. And actually, my degree is in kinesiology because you couldn't get a degree in neuroscience. Those came a bit later. And so I did my degree in kinesiology with a real interest in trying to understand how when you had strokes in different brain regions, what were the patterns of change that we saw in terms of learning for those patients afterwards? And then after I finished my PhD, I was able to undertake advanced training in brain mapping using fMRI. And I added in brain stimulation as a young assistant professor. And now I kind of put all of those approaches together in my lab now, many years later. So it's been a long process, but it's been absolutely fascinating and fun to kind of be at the front of a kind of a new and emerging field, because this honestly wasn't a field 20 years ago.
Definitely. And your research, from what I was reading, focuses a lot on neuroplasticity and motor learning. And so how would you describe the core mission of your work?
Sure. So I'm really interested in leveraging change in the brain in order to promote learning and then applying that to both patients and healthy people too, honestly. So when I was a grad student was when the first kind of demonstrations of plasticity in the brain came out. It was really, really exciting. But this idea that you can basically change brain structure, function, chemistry, and then behavior based upon an intervention is kind of a thrilling idea. It really is. I know it sounds like really hyperbolic, but if you think about it, we're all changing our brains all day long, whether we're students or faculty. And we want to kind of understand those processes, understand how to amplify them, and then use them to help patients get better after they've hurt their brain. So that's kind of how I put it all together. As we've done more work in the field, what's become really obvious is that there's a huge amount of variability from person to person, whether you're a patient or not, whether you're a healthy control, whether you're young or old, whether you have a stroke. Our brains just change really differently from person to person. And so that's become a real acute interest of mine and of my labs. And so in addition to kind of looking at learning and neuroplasticity, we now also do a lot of work trying to identify biomarkers. So that would be something that we can measure that might allow us to create subpopulations of people that will respond similarly to an intervention, for example. And so that kind of dovetails in with all of this. And it becomes really important if you think about like a group of people with stroke. Stroke is not a homogeneous disease. It's very different from person to person. It depends on a huge number of factors, sex, how fit you were when you had your stroke, what kind of health care you got, where the stroke is, your past history of movement. And so what we try to do is group those people into subpopulations and then find the right intervention for the right group. So that's kind of all of our work, all of my lab's work, mine and my graduate students and fellows, kind of in a nutshell.
Interesting. And so I'm really interested in what you're saying about how everyone responds differently. And so some of those are those the biomarkers that you were talking about, like looking at how fit they were, their age, sex, and those kind of things?
Yeah, so those are kind of some of the factors that help us subgroup people. The biomarkers that we're really interested in are biomarkers that will are either related to residual capacity of the brain. So you have a stroke, you have damage in one particular region. What brain regions are left over that can kind of take over some of that lost function? And so an example of that would be, we have a large study of people with very severe stroke. So people with very severe stroke, typically it means that their corticospinal tract, the long descending motor tracts of the brain, are really pretty much damaged to an extent that they're non-functional. And so those patients have often been thought of as not having much potential for recovery. But we know just from kind of observation that sometimes people actually do much better than you would think, considering how much brain damage they have. So we started searching for a biomarker that would tell us who would be the people with this severe stroke who might have potential to recover and should go to rehab versus people who have less potential. And maybe for them, you want to have a different kind of rehab, something where you're just helping them be more adapted to kind of this new reality. And so through that amount of work, we actually discovered that what's very important is the residual integrity of the corpus callosum, so the connectin, the big white matter bundle that connects the two hemispheres. In people with severe stroke who still have a pretty intact corpus callosum, they actually show a lot of potential for recovery. And so having that knowledge can have us talk to the healthcare system and say, hey, in these folks, they should still go for very aggressive rehab. They actually have a lot of capacity for recovery. And then other folks, other populations who don't, their damage is so extensive, they don't actually have that kind of compensatory route available to them. They're going to need a whole different kind of therapy altogether. So that would be one example of a biomarker. Some of the other ones we use actually are more neurophysiological, and we can derive them by mapping the brain using transcranial magnetic stimulation. So you can look at patterns of excitability in the motor cortex, and then you can predict whether or not, for example, someone's going to be a positive responder to an intervention like exercise. And could you use exercise to kind of promote recovery after stroke? And so patterns of cortical excitability and inhibition will actually seem to be linked in that direction as well. So those are just a few examples of the ones that we've worked on.
Wow, that's fascinating. So it seems like you guys are really trying to personalize the approach to recovery based on which parts of the brain are intact, which parts of the brain have been damaged. And really interesting what you said about that the people with an intact corpus callosum can recover better. And do you think maybe that has something to do with the other hemisphere kind of taking some responsibility?
Yeah, I think that you're exactly right. So you got there really fast. So yeah, we think that what the corpus callosum allows is for the ipsilateral hemisphere to assume some of that lost function, probably by activating things like the reticulospinal tract. You can look at the cingulate motor areas, actually have some bilateral connectivity. And so it's not perfect, and you're not going to have people going back to their normal function or function pre-stroke, but it gives them some capacity for a pretty substantial amount of recovery. What's even more important is if you ask the patient, how do you feel about your recovery? They will rate their recovery much higher. They're much more satisfied with it. And so that's another important part, is it's not just we see it with our, all of our fancy measures and clinical scales. We actually have the patient being able to articulate that this is meaningful recovery for them.
Wow, that's amazing. And I wanted to ask you, so you're a world expert on neuroplasticity, and I know neuroplasticity is such a big buzzword nowadays. And what do you think are some of the biggest misconceptions people have about neuroplasticity?
Oh, that's a wonderful question. Well, I think that people always assume it's a good thing, and it's just plasticity. Your brain just changes, and it just changes depending on whatever it is you're up to. So I think we forget that there are things like maladaptive plasticity where we see our patients do this. They move in strange ways that maybe aren't their most efficient or best for their joints, but they've kind of established a learned movement, and so they get kind of stuck there. And then, of course, all of us are stuck in different ways with different kinds of maladaptive plasticity, be it, my highly persistent caffeine addiction, right? So that's just a maladaptive form of plasticity, but also you think about pain and addiction as being other, really important elements of that. So I would think that would be the first one. I think the other misconception about plasticity is that people don't always know that it exists, right? So firstly, do you understand that you can change your brain? And then I think people maybe think that changing your brain is easier than it is. It actually takes a lot of practice. And that's something else we've looked at in my lab. So, we do motor learning studies where patients will practice something, or healthy controls too, 10,000 times is one of our studies right now. You make 10,000 of these reaching movements. And then we start to see change in both behavior and brain structure and function. So that's a lot of practice, right? That really takes a lot of persistence. And I think that's also something that we fail to recognize, that to really form a new habit and make it a very easily accessible thing for us, you're going to do a lot of it.
10,000 times. Wow. That's a lot more than you'd expect. I mean, you always hear this, people say if you do something 7 times, it'll become a habit, but you're saying it really depends. No. It might be a lot more than that.
It could be a lot more. And, 10,000 is not the magic number either.
Right,
this is another one of those variable things. Like it might, maybe you really good and you're younger than I am and you're really motivated to learn something. Maybe you can learn it in 4,000 times and it's going to take me 15. So this is something else we've been really interested in. So we created these a few years ago. One of my graduate students, I should say her name because this is her work, Katie Wadden. She's a professor at Memorial University now. She generated these exponential equations that we use in my lab where we can calculate individual trajectories of change. So we can look at how fast someone's changing early in practice, and we can actually make a prediction that turns out to be pretty close on of how much practice they need before they stop changing. So they asymptote. So think about a learning curve comes down, it gets flat. And doing that, we can really look at this crazy variability of how much practice to actually change brain or change behavior. Again, it's highly variable from person to person and task to task. But I think the take-home message is it takes more than you think. But I don't know what the magic number is. I wish I did. But I'm always we create an experiment. The hardest conversation we have is how much does someone practice? And we honestly just guess. Now I feel like for years we underdosed people. They didn't do enough. And now I think we probably are leaning towards the overdose side. So 10,000 is our current number, but that could be way too much. I really don't know.
Wow, it's super interesting. And I found it fascinating that you guys can actually predict that number using that asymptote early on to see how many times it will actually take. That's pretty neat, and I guess it goes to show how different it is for everyone. And exactly. Kind of going along with these lines, I know that a lot of people are under the impression that either there is no neuroplasticity once you get past a certain age, or that it's very hard to have that neuroplasticity. So I wanted to ask you, is it true that neuroplasticity declines with age? And if we still have that neuroplasticity as we age, how can older adults still reshape their brains effectively?
Yeah, that's a great question. So it's not true that we lose the capacity. We do, and we know that we do lose a bit of ability for learning with aging, but it turns out to be a really complicated story. Part of it is as we get older, our brains become less excitable. So the range of how much excitation and inhibition you can evoke seems to become a narrower bandwidth. That's a massive generalization, that's a massive average. Now I'm going to tell you that there's a huge amount of variability in that. seems to be really related to lifestyle. So people who maintain a high level of social engagement, of like intellectual engagement, exercise, they can continue to facilitate a huge amount of plasticity in their brain. So there's kind of these, we talk about them as being the adjunct interventions that you can use to continue to facilitate your capacity for learning. But there is some inevitable change with aging, and I hate to tell you guys this, but you're already on the downslope, even at your age, of how much change you can make in your brain if you think back to being a young child and thinking about kids who grow up learning to speak multiple languages, for example, or all the things you learn through what's now called middle childhood, which is about out to age 8 or 10. I mean, you're learning just an amazing amount of information at a catastrophic speed, right? Everything from learning to walk, to run, to ride a bike, to ski, to different languages, to reading and mathematics. I mean, those are crazy neuroplastic tasks. So we actually think that the cortex has done most of its remodeling by about age 12 or 14, and you're pretty much having a cortical thickness. Yeah. And then the long white matter tracts of the brain become fully mature at between age 20 and 25. And males are later on both of those things. So males mature much later for the white matter tracts, and cortex probably closer to 14 than 12. But it's early. It happens early. And then after that, the changes that we make are less huge, but they're still highly relevant, and they continue to persist really as long as you keep trying to learn something new.
Interesting. I mean, it's the cliche, but you can definitely tell that the males maybe mature a little bit later. I mean, I'm thinking back at some of the decisions I've made in high school, or even still some of the decisions I make, I understand. But super interesting. It
explains a lot about all of our 20s, I think. Mine too. I wasn't exactly focused.
For sure. No, it definitely it ties things together a little bit. But in terms of that neuroplasticity, I think it seems like it all brings back to this lifestyle point, and it's like use it or lose it. If you're telling your brain's going to adapt, it's like you're telling your brain you need to keep this plasticity because you need it. You're still engaged socially, engaged intellectually, using your muscles, firing those neurons. It seems like it's really your brain will take what you give it almost.
Yeah, I think that's exactly, it's exactly the use it or lose it principle, and the same as we would talk about like for muscles or even bone density.
Right.
And it's exactly the same. And our brains get better at becoming adaptive in things that, we work on and think about. So like I'm really good at neurophysiology, right? But if you came to me with, I don't know, something about acidity in the oceans, I would probably be a bit lost. I know I could probably figure it out, but I'm not 100% sure. So, I mean, we become very good at what we're good at, if that makes sense, as we focus in different areas. And that's just a result of that plasticity kind of leveraging on top of itself and spinning up.
Totally. And this might be a bit of a stretch, but if let's say you have a mastery in one area and then you want to go and master another area, is that going to take away those connections that you made in that past area?
Yeah, it's really interesting. This is this idea of like cortical competition, like how much room do we have in our brains for certain things? I don't know that it's interesting. I don't know that it's a competition, but we do only have so many neural resources, right? And so we do, at least in motor skills, there's really good experimental evidence of what we call interference effects. So the one I always talk about with students in the classroom is things like, if you're a tennis player and someone hands you a racquetball racket, you're going to be really messed up, right? Because you have something you've learned that is one thing, and we just changed it enough to kind of screw you up, if you will. So that can happen. On the other hand, if the things you're learning are complementary, you may see, a faster kind of change as you learn the second thing. So I think it depends a little bit. People have studied this a little bit in the language domain. Say you learn, like, you're a good Canadian and you can speak English and French. Well, your French is probably going to really then help you also learn Spanish and Italian because they're similar. They're different, but they're similar kind of things. On the other hand, being bilingual in French and English may not help you with, Mandarin or something, even German, something even farther away in a language domain. So that kind of gives you kind of a sense of it could help, but it also could hurt quite a bit. I always think, though, learning how to learn something new, just the process of learning it, no matter what it is, I always think that then helps you learn something else that is new. Like, okay, I know this is going to be hard and I'm going to make mistakes, and as I practice, it'll get easier and easier and easier. And so I think that there is that ability as well that transfers across skills.
Well, that's great news. That's fascinating that if once you learn something, that'll help you to learn more things. I mean, that's I feel like I feel optimistic when I hear that. It's like the more things I'm trying out, the more things I can get better at. It's really interesting how you're saying, like, sometimes it will take away, but sometimes it can also complement if it's similar enough.
Right.
And I kind of want to dive a little bit closer into the work you're doing now. So your research more focuses on the motor learning and recovery after stroke. So what have been the most surprising findings about how the brain adapts after injury?
Yeah, that's a great question. So the field has really changed over the years. I think that the amount of plasticity we can see in the brain has been surprising. And so this may not be as surprising to you, but really only about, 10 or 12 years ago, the big thinking in the field was that all of the recovery we would see was related to kind of functional reorganization in the brain. So the way the brain was activating would shift to accommodate the damage. So that we were seeing it through functional MRI. And we thought that, I think, because we were all doing functional MRI and that was the tool that we had. And then really led by the physics group at UBC, we began to be able to do things like image the long white matter tracts of the brain. And so we could then understand that their damage was definitely related to poorer clinical outcomes. And we thought that they really could never change, that white matter damage and the white matter tracts of the brain just didn't change. We had a big technological advance where now we can actually image the thickness of myelin as it wraps those long white matter tracts. And so it turns out that a huge new thing that's been so exciting and was shocking to me when we first started learning this is that our oligodendrocytes in the brain, which make myelin, this insulation along these axons, these cells are massively plastic. They're actually adapting all the time and in real time. So they're getting thicker, they're getting wider so that the nodes of Ranvier are farther apart, and so things can go faster. They're shrinking to slow things down. And we now really think about them as being kind of the gatekeepers for the timing of how electrical impulses are traveling across this really complicated system of our brain. And that amount of plasticity is not only being heavily related to learning and to recovery after stroke, recovery after spinal cord injury, but we can actually image it to a certain degree using a technique called myelin water imaging, which was invented here at UBC by Alex McKay, who used to be the director of the MRI Center, and Corey Lally, who is now our new director, was working with him as a student at that time. And they have developed all these tools and techniques that now 10 years later allow us to take these really elegant looks at how after you have a lesion in the brain, we see functional reorganization around it, but we also see these structural changes very distant from it that are helping to accommodate for that specific lesion. So in terms of like a neurophysiologic standpoint, it's been so exciting to understand this massive capacity of change across the whole brain, and then start to understand and unpack how that changes from person to person and with different interventions. And so that was the biggest surprise to me in the last few years, was how dynamic our white matter tracts are. We always just thought of them like, when I was a student, they were just structure. Like, it was like no more exciting than the walls around you. And now we realize those walls are moving, all the time. It's pretty neat.
Wow, that's really fascinating. And I had no idea how much of that imaging was developed here at UBC. That's really crazy that you're able to see the changes in the myelination of the oligodendrocytes. And I was hearing about it in certain papers, but you put it really well there. I mean, it's fascinating, this new research coming out, that how the oligos are actually so dynamic and really facilitating those changes. And it's almost like I never realized about the shrinking and lengthening of those nodes of Ranvier. It's almost like adding, if you think of it like an electrical circuit, like adding resistors.
Exactly. It's exactly what it's doing. You nailed it. And so imagine that happening like in real time. We first started looking at this, we thought that would take a massive amount of practice. That's where our 10,000 repetitions comes from, right? Because I was like, oh, it's going to be so slow. We need tons of practice before we see that in a person. It now turns out that it's in real time. And these are not experiments from UBC. There's some groups in the US actually doing these experiments where they have oligodendrocytes like under electron microscope, and they can make these movies of them. They're changing in real time as impulses are being transmitted across a nerve. So it's really, really exciting. It now turns out, this is really new, that they also just discovered that so you have an oligodendrocyte, and it has a whole bunch of dendritic branches. And so one oligodendrocyte is actually touching multiple individual axons. And so it can, by just influencing one of these cells, you can influence the myelination of multiple axons. So it's really neat. And they're all kind of turning on and off kind of in concert with one another. It's a very elegant system. Wow.
That's so fascinating. I mean, more and more research is just showing how important the glia are for so many different functions in our brain.
That's the other one that I'm really excited about, although I can't image glia.
Yeah.
And so I don't have as much understanding of it in a human, but it has been, I was taught they were the 2x4s of the brain when I was a student. Right?
Totally.
Not quite.
It's all dynamic. I mean, nothing in biology is simple like you think it's going to be. It's all super complex. But I mean, the more we learn about it, the more strategies we have to help people with relearning these behaviors. And on that note, you touched on this a little bit earlier with the biomarkers, but how close are we to being able to personalize a stroke rehabilitation based on an individual's brain imaging data?
Yeah, I think that we could really start to do it. The problem is most patients don't get brain imaging data. So we have this gap of translation. And it also then depends on the subpopulation. For a severe patient, for a person with a severe stroke, I think that it's critical that we have this imaging and we understand how much is left in those long white matter tracts of their brain, the corpus callosum, the corticospinal tract, the reticulospinal tract. And so that's the work that we're doing. We have a prospective longitudinal cohort study running right now. We have 4 patients left to enroll in it. And what we've done is we've taken folks who have these types of severe strokes. We recruit them at Vancouver General. We follow them for a whole year after their stroke, and we image them multiple times. We collect motor and cognitive data. We collect TMS data. And we collect MRIs. And so what we're really hoping to do there is we already have a kind of decision tree, an algorithm, if you will, of decisions about the data that comes out in terms of mapping their brain and what group we think they'll be in. So we're going to use these data now to test that model and understand if our predictions are correct. And if they are, then that would be a first really essential step then to go back to health authorities and say, here's a tool you can use to decide who and when people should have therapy, and so to personalize it that way. So that's a huge step forward in that arena. Other groups are using different kinds of imaging and stimulation to do similar work to try to personalize outcome. There's a bunch of work around looking at brain stimulation and trying to understand how we can drive more outcome using it, and then who will be the people who will respond to that. And we're part of a large network across Canada called CanStim. We have 13 labs that are harmonized with the same equipment, all running a clinical trial right now together, with UBC being one of them, based out of GF Strong Hospital, where we're all taking acute stroke patients, giving the same intervention, and then at the end of that, we can look back at responders and non-responders and take a look at some of the biomarkers we've collected, and then try again to say, okay, we're going to personalize who gets this type of treatment based upon these early things that we can measure. So I feel like we're like right at a tipping point, like really close to kind of tipping it over the edge and starting to really implement these things. So it's actually a really exciting time to be working in the field, at least in my opinion.
No kidding. And I mean, the thing I wanted to touch on. I mean, it sounds like right now you're in a point, and with these other labs, where you're getting so much data. And I know that AI and machine learning are becoming increasingly prevalent in neuroscience. So do you see that these technologies could help shape the future of stroke rehabilitation?
Yeah, I think they could be used to like implement these algorithms and then probably refine them as well. Because, the algorithm we've designed is just limited by our imagination, right? And so as we have more data, we could take that as a scaffold algorithm and ask AI to refine it. Like, what other things should go into that might help us make these decisions? I think the real limitation we've had so far is that we just don't have enough data. And that's why these consortiums like CanStim are so important, where instead of one or two labs doing it, we have 13 across an entire country, massive variability. A stroke patient in Vancouver looks very different than a stroke patient in Newfoundland.
Right.
People here are oddly fit, even when they do have a stroke. We're older, we're more fit to start. And so it's a different pattern. It may not be reflective of all of Canada.
Right.
So I think as we get more and more of these databases together, we're going to really start to exploit the machine learning aspects of it. We already are using some machine learning in my lab, and we're using it to extract kinematics. So where's the arm moving in space without markers? So you could basically, I could start reaching on this call. We could take the video recording from it, run it through we were using a program called MediaPipe, and it would extract out all of the ways that I move my arm, did my trunk move, and give us these really beautiful joint angle data. And so we actually are using that in the lab because it's so much cheaper and easier than having a motion capture system to help us understand how stroke arms are moving and how then that changes with an intervention. So we're definitely on the edge of machine learning, I think, being a really core part of a lot of what we do.
Fascinating. I think it seems like just in every field, it can be leveraged in a way that just helps to speed things up. And I think that as we get more data, like that joint study that you guys are doing, it could be really interesting to envision a future where you get your imaging done. We have all these markers of stroke, severity and the different places that were effective, and then you have an algorithm that gives the patient the best rehabilitation strategy based on that data. Do you think that's something that could happen in the future?
I do. I really do. And the exciting thing about that too is that we could do these things remotely. So even today, we're both in Vancouver, but we did this online because we're embarrassing Canadians and it's snowing a little bit, and so I'm not at UBC, right?
Yeah.
But the bigger problem is, our patients who are really remote on the island or up north, even out to the east. And so, those people really don't have access to very good rehabilitation care. They have to travel distances. There's not a lot of specialized people to help them where they live. So imagine if we could do a lot of this virtually and still get excellent data to really be able to help shape a rehabilitation program. That would be really quite transformative for Canada. And I do a lot of work with colleagues in Australia, and they actually have the exact same problem because Australia is huge like Canada, and everyone lives in a few small places, and then people scattered through the middle and up the coast. And so we've been working with some Australian colleagues quite a bit on these kinds of problems of how do we provide better care to remote and rural areas. So I do think, and I hope that that's coming probably sooner than we think. AI just seems to move faster than we can I can't keep up anymore. It's wild. And so it's probably going to happen very soon.
Wow. Well, that's very exciting to hear. And I think everyone listening to this will be super impressed with all the work that's being done. And thank you for helping to pave the way for that. I think it will be a really valuable tool for a lot of Canadians and Australians as well, and people around the globe. Once we get enough data and once those algorithms start to come online. So now I'm going to transition a little bit into our closing section. So if you could investigate any question in neuroscience without constraints, what would it be?
Oh, wow. Gosh, I have so many questions. I don't even know where to start. That's wild. Well, I'm sure I'll be able to do this. This is maybe not as big as you might think, but it's something I'm just absolutely fascinated with right now. I don't feel like we understand if and how neuroplasticity takes place in the spinal cord. So what is the cord doing? And why are we so bad at helping it get better when it's damaged? So stroke is 1,000 times easier than spinal cord recovery, right? We don't see much neuroplasticity. It's a devastating injury when it does happen. But kind of at a more basic level, we must be having reorganization and neuroplastic change in the cord and the different motor neuron pools and probably along the long white matter axons. And we just don't have a way to interact with the cord, mostly because it's so well protected by the spinal column. It's really small for our MRIs. And so that is something that I just think about a lot. We're trying to do a little bit of spinal cord imaging right now, but it's so messy. And so this is something that I would love to get into a little bit more. Can I have two?
Yeah, of course.
Okay, I'm going to be greedy. The other area that I'm really thinking about that we've not done a good job on with stroke, if you can believe it, is trying to look at blood-based biomarkers. So markers of inflammation and plasticity markers. And so that's something that I think is much more accessible, but I would love to see happen. If I just had a pot of money, this is something I would dive right into. And we actually have the expertise at UBC to probably do this through the Wellington lab. So it's something Cheryl and I have talked about a few times, but we just haven't been able to kind of generate the funding. And there's so little data out there that I'm not even sure where to start. We need to just do something crazy exploratory. But that would be the other area of science, of neuroscience, as it relates to stroke recovery, that I think could be really transformative. And those are also data that we need to put into these AI models that you and I were kind of just spitballing about. It would really make a big difference. So Those are my two wish list items at the moment.
Yeah, thank you for those. And I guess I'll start with the blood analysis. So you're saying that in addition to these neuroimaging techniques we have, that if we had some ways of measuring like brain inflammation and growth factors, et cetera, in your blood from just like a simple blood test kind of thing?
Yeah, that's the idea, to ask it first. It's always so tricky because, what's in the peripheral blood may or may not be what's in the brain, right?
Right.
The blood-brain barrier is really good at doing its job. But it would certainly be a place to start. So are there peripheral markers? There's been an idea in spinal cord rehab for a while that you really shouldn't intervene until inflammation recedes a certain amount. And so I know of like one trial in the US, it's a go, no-go trial. So they keep testing spinal cord. They keep testing peripheral inflammatory markers. When they go below a certain level, then they have aggressive rehab intervene. So something like that, but with stroke maybe.
Okay.
We don't know how early we can intervene. We know that there are some good data that if you intervene too early, you can make the stroke worse, or you could actually kill the patient. It has resulted in death. There was a huge study a few years ago called AVERT that showed that you really shouldn't intervene too early, but we don't know what that means. And even within that population of people, some died, some got worse, but some got better. And so the question is, what is the marker that's going to tell us that it's time? Right? So you can get in before maladaptive plasticity takes place and really start to shape the brain from the get-go as early as you can.
Right.
So that would be the idea of some of that blood work, is could we get something in the peripheral blood that would tell us that information? And I don't know. I really don't know. Something I've thought about, and I would love to have a bit more data to have some ideas and be a bit more hypothesis driven around it.
Yeah, and I wonder, even with with TBIs as well, do you think there would be even possibly some crossover with certain markers?
I'm sure that there would be, and I would let the TBI data inform these questions. And so the Wellington Lab is doing some work with TBI and looking at starting to extract some of these inflammatory measures. And you do see them go up, and they do track with severity. And so the question would be, is that also translate into stroke?
Okay.
And I really don't know. Stroke's so different because it's really a disease that largely happens to older folks, whereas TBI tends to happen, not always, but it's a younger group, so you don't know if you're going to see the kind of same patterns, but it's definitely a place to start.
Okay, super interesting. Thank you for those answers. Very insightful.
Yeah.
And just one last final question. What advice would you give to young neuroscientists looking to make a meaningful impact in the field?
Oh, yeah. So just pursue your questions, right? If something seems fascinating to you, like, go for it. There's so much more we don't know than we do know. I mean, I would say there's like no wrong questions. There's no silly questions. And so I think just kind of find an area that you really are fascinated by and dive into it. The other thing that has really happened to me in my career is the tools and technology we've had access to are light years different now than when I started. And so you may want to study something and think, oh, we can't image the spinal cord yet. That's usually not the case. You might not be able to do things now, but the technology is just improving so quickly, and we're learning so much so fast that I really think that if there's something you're fascinated in, go for it, and the tools will come.
Fantastic. Well, everyone, you heard it from Dr. Boyd. Go look for those questions that really spark your interest, and make use of all the cool new tools that are available and continue to be developed. So, Dr. Boyd, it's been a pleasure. We went through a lot of really interesting topics. I learned a ton today, and I really want to thank you for your time being able to do this.
Thank you so much. It's been really fun.